Welcome to the Soft Matter Show. This is Amal Narayanan. I'm a chemist and a polymer scientist. In this podcast, we will talk about soft matter science and have fireside chats with the researchers. Please enjoy the show. In this conversation, Dr. Anirban Sengupta joined us to chat about synthetic platelet-inspired technologies for blood clotting. We dig deep into his transformation from a chemist, chemical engineer, to a platelet biologist. The conversation explores the futuristic technologies that could be used in critical medical care and in civilian and combat wounds. Later, the conversation led to the philosophy of translational biomedical research, cross-disciplinary collaborations, and persevering in the academic journey. The conversation was so much fun and is packed with lessons for early career researchers and entrepreneurs. Let's hear from Anirban. Today we have Dr. Anirban Sengupta on the Soft Matter Show. Anirban is a professor in the Department of Biomedical Engineering at the Case Western Reserve University and is a co-founder and CSO of Haima Therapeutics. He is a prolific researcher in the field of translational biomedical research. He has published over 60 peer-reviewed publications and owns several US patents. He has raised millions of dollars as research funding, and he is an engaging entrepreneur. Let's hear from Anirban. Welcome to the Soft Matter Show, Anirban. I'm so excited to have you here today.
Thank you, Amal. Thank you for the invitation. And I'm really excited to have this conversation about, I guess, philosophical and scientific aspects of our research. And I'm looking forward to where this conversation goes.
Yeah, thank you. So the first question I have is like sort of a very internal information that I got from some of my friends in Case Western Reserve University. So you are known as the platelet guy in Case Western Reserve University, and I think even in the biomedical research arena. So do you happen to know why and how that happened over your career?
Yeah, I mean, I can only guess, right? I never call myself a platelet guy myself. But I can, and I don't think I am the platelet guy. I can name you like 10 really the platelet guys and girls. Or men and women in this field. But I think the terminology stems from what I remember many years ago. I think I would say at least 8 or 10 years ago. We were talking with a group of people at a conference, probably a Biomedical Engineering Society conference, and one of my former PhD graduates was really enthusiastic about me opening a Twitter account. I did not have a Twitter account at that time. And I'm like, I don't know what Twitter is. I don't even know what to do. And she said, so her name is Elisa Master. She is a prolific researcher, and she's at a company called Nucleus Biologics in San Diego, VP of research. And she said, you know what? I will open a Twitter account for you, and I will make your handle the platelet guy. And I'm like, that sounds really weird, but hey, do your thing. But I think the reason that this terminology kind of came up, especially within the Case Western framework, is that I would say about 75% of our research is inspired in some way by blood platelets. And it spans from, making artificial platelets to all the way using platelets as a paradigm for drug delivery and wound healing. And so there are probably 5 or 6 parallel NIH and Department of Defense funded research projects, all of which in some way or form delve into platelets' natural mechanisms and then either leverages it or mimics it or prevents it or promotes it. So there are all these different nuances that are centered around blood platelets that we do in the lab. And so thematically, I have become the platelet guy. Okay.
So you did not start as a platelet guy, right? So you started your research more like I think you worked on some tyrosine-based materials which was then used in some biomedical applications, if
Right.
I'm thinking it correctly. Then
Right.
you moved to the platelet research, or like more of a blood related research. How did that
Right.
transition happen?
Right. Yeah, you're absolutely correct. And I heard you say that you're going to have Professor Cohn over for a conversation at some point in near future. And so, my PhD research was actually inspired by some of the research that Professor Cohn was doing, and Dr. Joy was actually probably involved in that at some point. And Professor Cohn's research had delved specifically into polycarbonates and polyarylates using amino acid systems, but non-amide linkage of amino acids using their reactive pendant groups. And that research, when I was a PhD biomaterials researcher, but in the chemical engineering department at Akron, that research actually fascinated me because my undergraduate training is a dual degree in chemistry and chemical engineering, and I saw the materials engineering as sort of this interface of both chemistry and chemical engineering. And I delved into that field of research, but focused more on polyphosphates and polyurethanes. And so that's where the tyrosine and serine and all these other amino acids that have pendant groups that you can react in other non-amide ways came into existence. And then, there's a library of polymers were created by virtue of that research. Obviously, the carbonates and arylates have gone into clinical translation. The phosphates and urethanes are still in preclinical research, and different people like Young Yeon at Akron are using that for drug delivery and all sorts of stuff. My transition into blood research was actually through my postdoctoral training at Case Western. So I joined Case Western back in 2003, fall of 2003, as a postdoctoral researcher. Really, in a strange way, looking at materials designed for coating blood contacting devices to prevent clotting. I did not know much about the complexity of clot biology. Obviously, I knew that blood clots and platelets are somehow involved in it. That you learn in high school biology, but it's a really elegant and complex process, and I did not have much expertise and information on it. But due to the research that I started more as a polymer engineering person to make brush polymers that are inspired by the endothelial cell glycocalyx to prevent clotting, I started reading about clotting. And I was hooked because it's like, if you're an engineer and you look at clotting, it's like this integrated system of 13 or 14 differential equations. If you're a biologist who is looking at clotting, it's all these coupled and decoupled enzyme kinetics interactions. If you're looking at a biomolecular or a cell biology person, you look at all these sort of cellular and molecular amplifiers and inhibitors of clotting. So there is something for everyone in that clot biology subset, and it really, really hooked me onto this context of how elegant nature is, and then allowed me to get inspired by bits and pieces of that process to create technologies to either promote or prevent or leverage it. So it was kind of a transition through self-learning. And then as I started learning, I obviously realized that, this is an area that because I didn't have any background in, I need to start learning from other people who have spent their lifetime learning some of these things. So I was very active, and I'm still very active in building collaborations with people in the clotting and coagulation biology field, from whom I learned more and more. And it was sort of a win-win because it allowed us to team up as engineers and biologists working together towards technologies, right? And so it was sort of a bidirectional enhancement of knowledge and achievements.
So if I dig deep into that, whatever you just said about the learning curve of you transitioning from a chemical engineer to more of a biology related or like a blood related researcher. So one of the things I want to ask you is like, who were some of the people or the groups that helped you in transitioning this or this into this more of a blood related research? Like which groups do you follow in terms of that, or which resources did you go towards when you started looking at new research?
Sure. Yeah. So, I mean, from a polymer or a materials engineering standpoint, obviously my background and my sort of, I owe it to University of Akron because, under the mentorship of Stephanie Lupina, Jim Harwood, and others, both in, chemical engineering and polymer science and engineering, that's where the whole context of not just designing new materials, but designing it efficiently and thinking about process development, that kind of sort of translational aspects, specifically focused on materials design, that kind of emerged out of my training in Akron. But in the blood-related field, there are many people I owe in terms of getting me to the point where I am now. One of the first people I spoke with is Professor Candice Marchant, who was the former chair of pathology at Cleveland Clinic. She still serves as a clinician at Cleveland Clinic, but not in that role. And she was very well versed in coagulation biology. Dr. Keith McCree, he's the director of benign hematology at Cleveland Clinic, with whom I have had collaborations for a long time. And then Dr. Marvin Neiman, who is in the pharmacology department at Case Western. So these were some of the early sort of collaborators, influencers, bounce of ideas kind of people that I worked with. And then as my research sort of expanded, the folks that I have spoken with the most are Dr. Wolfgang Bergmeyer, who is at UNC Chapel Hill, Dr. Joseph Italiano, who is at Harvard, Dr. Paul Kim, who is at Tari, Canada, Dr. Matthew Neal, who is at University of Pittsburgh, Dr. Philip Spinella, Washington University, St. Louis, Dr. Wei Li, who was at Cleveland Clinic, but now at Marshall University. So all these names that I'm saying, these are names that, I look up to when I need to learn new things. Dr. Elisa Wolberg, who is also at UNC Chapel Hill. So they are my knowledge banks, but also these are people who I collaborate with in terms of, maybe there is an engineering aspect that they want to know. Maybe there is a biology aspect I want to know, and we kind of meet somewhere and exchange of ideas lead to new research projects, new grants, and so on and so forth. And then in very recent years, folks like Dr. Evi Stavru, who is at Case Western and at the VA hospital in Cleveland, who doesn't study platelets, but who studies neutrophils. And so collaboration with her has allowed us to kind of evolve into just platelet-related research to platelets and other blood cells, like platelets and neutrophils, platelets and red blood cells, platelets, So there are diseases where it's not a single cell scenario, but it's a heterotypic multi-cell scenario. And now, over the last, I would say, 4 years or so, our research has expanded into some of those disease areas. So it's a constant learning environment that builds through collaboration, and it also builds through the ability for all sides to kind of approach this process as a blank slate. That, okay, I don't know anything, tell me about it. And then the ideas kind of take shape by themselves.
Yeah, that is interesting because your approach has always been very collaborative. Your works has been extremely collaborative. So as you mentioned in the previous part, like you said that you joined Case Western in 2003, and you have been there since. So that is like 18 years of your life has been in Case Western or Surrey University. So one of my question is that what are some of the highlighting projects or the tasks that you are involved with which keeps you awake in the middle of the night?
I would say all of them. No.
That's like any other scientist. They are favorite childs. All of them are
Right.
their favorite childs.
Right. So I think what actually keeps me awake or gets me super duper excited is not just the science of things, but actually the ability to translate. I think that's a very important thing to consider if you want to do translational research. It doesn't mean that everybody needs to do or wants to do translational research, but that is something that I feel very passionate about. So that's why I'm putting an emphasis on it. And what that essentially means is that, there is cool science and then there is useful science. Okay. And I kind of make this distinction oftentimes, right? You could do a lot of really elegant, and this is not undermining the cool science part without the translation. I think every science starts at the discovery phase, right? And there's like a lot of discovery and complications and complexities that get recognized and evaluated and all sorts of stuff. But at some point, one has to kind of step back and look at this volume of work, and then really think that, okay, do I want to focus on this volume of work and have 30 Nature papers and whatnot, and that's how I want to finish up my career? Or do I want to look at, I still want to do all this discovery research, but do I want to look at bits and pieces of that volume of work and see if I can realistically move the needle a little bit more towards the patient, right? And that's the translational part of it. And the reason I'm making this distinction is that the more complex your technology is, the more difficult it is to translate. It might become easier to put it in a science or a Nature paper because it has all sorts of things and complexities and 8 different in vivo models and whatnot. But it doesn't necessarily mean that the product of that research could go into a patient. And that's fine, because that discovery is very valuable. But where I have kind of made my passion move, or if you, as you asked, what I think about the most or keeps me awake at night or whatnot, is identifying pieces of the volume of work that we do in the lab that has a realistic chance of moving to that next phase of translation. And that's what I constantly think about. And it is, I would admit that it is sometimes detrimental, because sometimes I would sort of overlook some really cool science and get distracted by the translation end of things. But I always try to come back. Like, I don't just throw it away. I always try to come back. And so thinking the spectrum of basic science to translational technology advancement is what gets me the most excited, right? The whole spectrum of that. And it's
So what are some of the examples of that? Like, can you give me one example from your lab?
Sure. So let's talk about, you started the conversation with the platelet guy. So let's talk about platelets for a second.
Yes.
So platelets as a cell, it's a very complex cell because it doesn't have a nucleus, which means, it doesn't have transcription translation end of things, but it contains a lot of mRNAs, it contains a lot of granule contents, so it's like loaded with 1,000 different things as it's being produced from its mother cell, the megakaryocytes in the bone marrow. And then as the platelets come into circulation, obviously the most well-known role of platelets is blood clotting, but platelets are also involved in a variety of signaling aspects and mechanisms and processes in inflammation, in cancer, thromboinflammation, which is sort of, your immune system and your coagulation system are interfacing into a disease like COVID. So COVID is one of the scenarios where the coagulation biology and the immune biology have sort of interface with each other to create the critical state of COVID patients. So, this is a rabbit hole that one can kind of go down into, that, what do platelets do and don't do? And it's overwhelming because there are thousands of papers regarding some of, all of these scenarios. And so when somebody says, including myself, that I'm going to mimic platelets, people look at you crazy. Like, what the hell do you mean you're going to mimic platelets, right? And so then it becomes important to convey the message that what I mean by mimicking platelets is that I don't want to mimic the complexity of biology, of all the signaling pathways, of all these things that are going on in platelets, but rather I want to focus on specific functional outputs of platelets and mimic those specific functional outputs that may be unique in one disease, but doesn't matter in another disease, so on and so forth. And that way you get what I call this sort of reductionist modular engineering, right? Like I always give this example that the airplane doesn't need to flap its wings to fly, although it's inspired by the bird flying, right? But you don't have to get into the complexity of all the muscle functions and the signals and aspects of the bird flying.
Yeah, that's an outstanding analogy.
Right. So you basically kind of, translate that over with engineering by putting the aerodynamics that you have mimicked, the liftoff that you have mimicked by your engine function or rotor function or whatever kind of plane you're using, and then your stability, your weight management, all this other stuff. So that's sort of the kind of approach that we took with platelets, and then we kind of chopped it up from a design standpoint. We chopped it up into, okay, these 3 or 4 functions are the most important if we want to impart clotting function. This other function is most important if we want to impart inflammatory signal. This other function is most important if we want to impart an immune response. So you kind of end up creating this menu card that now you can mix and match to create platelet-inspired systems that are uniquely designed for specific functions. And that becomes more useful than trying to mimic the whole platelet in all its elegance and all its complexity. That is, I wouldn't call it impossible because some brilliant person will do it, but that is not something that I can envision to be possible in a timeline that can also help it translate to the clinic. You see what I mean?
Yeah.
So that's, you could make an airplane that would fly like a bird flapping its wings, but the amount of complexity that would go into the design, it will not reach the sky.
Yeah, exactly.
Right?
So that is an exciting point. So like one of the things in that is like, can you give one specific example of that platelet-inspired system that you have developed in the lab?
Right, sure. So let's take at the most well-known role of platelets, which is clotting, right? The first response of platelets, there are many cascade responses after that, but the very first response of platelets in clotting is that it would recognize certain proteins that are being exposed at the bleeding or the injury site. They'll go stick to it, and then they'll start piling up on each other. So it's like sandbags trying to prevent a flood. So you're gonna download the sandbag there, and you're gonna stack it up to create this sandbag wall that would prevent the flood, right?
Which is the blood clot.
Which is the blood clot,
yes.
Right?
Yeah.
So if we look at the sticking and the aggregation as sort of the most critical first response, everything else kind of starts after that. There is a lot more happens in blood coagulation, but that it starts after that. So if we look at this platelet role of sticking and clustering at the injury site, we took those two functions and we mimicked them with lipopeptide or polymer peptide systems on nanoparticles or microparticles. And they mimicked just that part of platelets. And we found that just mimicking that part creates a booster effect in clotting. So we could create a clot maker by mimicking only a modular aspect of platelet function in clotting. And now that has emerged as a technology that's now being advanced further through the company that I started, right? So,
Yeah. So what are some of the implications of it? Like, what are some of the implications of this technology which allows you to control the clotting?
right. So you mean from
Real
like
life.
who would use it?
Yeah.
Where would it be used?
Yeah.
Sure. So the biggest use that we see is in treating heavy bleeding, hemorrhagic bleeding. Okay. So this can happen. I mean, it's very easy to grasp that this can happen in battlefield because there's war going on and gunshots and explosions and whatnot. But it is actually, if you look at the CDC chart, they call it unintentional injury or unintended injury. They have like this strange name to it.
Yeah, I think there are different types of accidents, so they have to classify it in that way.
Right. But the bottom line is that it's some traumatic injury. So it can happen from car accidents. It can happen from somebody was putting Christmas lights on their roof and fell over. It can happen in sports injuries, right? In recent years, unfortunately, a lot of mass casualty events, people gunning down people everywhere, stab wounds, all sorts of stuff. So from a civilian scenario and obviously from a military scenario, there is a huge number of patients that get these sort of heavy bleeding injuries, and they need to be treated by transfusion of blood products, including platelets. Now, real platelets are used for these transfusions so far. Clinically, right now, real platelets are used, okay? Real platelets are obviously isolated from blood donors. So your donor pool will control how much platelet you can get, which means you're already limited there. It's not like you have thousands of donors giving blood and platelets every day, right? Even in a pandemic scenario, it's even worse, right? There's barely any blood drives going on. So this is number one. Number two, platelets specifically have the shortest shelf life. Platelet shelf life is only about 5 days.
Why is that?
They get contaminated with bacteria, and they also get sort of activated, and they kind of shed all their good cocktail that they have inside them over time. So it's like food going bad kind of thing, right? So they just go bad after 3 to 5 days. Sometimes they can be used at about 7 days or so, but that's the max. After that, platelets cannot. So what does that mean? That essentially means you cannot stockpile these things. It's not like you have a big silo full of platelets and then you can release it on demand when needed, right? Third thing is, ambulatory challenges. Platelets cannot be carried out very easily to the roadside or to the battlefield because they need to be stored in very specialized bags. There's a lot of sort of logistical challenges of taking platelets outside a blood bank or outside a hospital. So when you look at combination of these factors, you end up with this big gap in real platelet transfusion. And it's evident by the fact that there are, so mostly funded by the Department of Defense, there is now research going on in cooling and freeze drying and lyophilizing real platelets with the idea that, okay, maybe we can increase the shelf life. Now, cooling and freeze drying and all this stuff, it does increase the shelf life to a certain extent, maybe, from 5 days to a few weeks. That can happen. But the flip side, the compromise there is that when you re-thaw or resuspend these cold or lyophilized platelets, you lose about 30 to 50% of their functional aspects. So you can store them longer, but you have a lesser performing product, which creates the opportunity to create a technology that works like platelets, but can sustain its functionality over long periods of time is fully synthetic, doesn't require type matching, doesn't require donor availability, can be stored as liquid or as freeze-dried powder in scaled up proportions, and can be taken outside in an EMS ambulance or a medevac helicopter or in the field for battlefield use and things like that. So suddenly you identify a big technological gap that can save lives, and you try to create a product that can address that gap while keeping in mind what are the challenges of the current products or current approaches, right? So this is sort of how we came up with the motivation, I would say, an inspiration to create a clot making system, a platelet-inspired clot making system, because our vision was, okay, if this system can translate to actual clinical use, one can imagine somebody in a car accident in some remote civilian area where there is no nearby access to a big trauma center or a big hospital, but the EMS can actually put in this product in the patient and stabilize the patient for long periods of time until the patient can get access, can be ambulated over, can be flown over to where real blood products are available. So it's kind of a bridge technology.
Yeah, that sounds like a very wonderful technique, because one of the reasons I read like recently that the reason for the death in the battlefield or even civilian polytrauma is that the time between they reach the ambulance and when they are in the site. So if there is anything in between technology that can be provided, that would be an exciting work. And like to just say the numbers, and it's a very commonly, like I think it's the second most cause of death in the United States in 2017, the unintentional injury. So the implications of a technology that can reduce this unintentional deaths, or it's called preventable deaths, is an exciting platform. I think that'll be very useful, I think, eventually.
Right.
So is that what the Haima Therapeutics are involved with? Is this what your company Haima Therapeutics involved with?
Yeah, I mean, that is, so Haima has, they are principally involved with this now. From a vision standpoint, Haima wants to delve into a variety of platelet-inspired technologies for various applications, but they are starting with this hemorrhage control, bleeding control technology as their first portfolio product, and then kind of build on that.
So how is the funding raised for both your research and at the Haima Therapeutics for the research in this field?
Sure. So obviously academic research from a biomedical engineering standpoint is the biggest source of funding is NIH. And so there are RO1, Research Award 1 grants that are anywhere between $2 to $4 million per grant, 5 years, 4 years, 5 years grant. So those are mostly the academic grants that we raise money through. The company currently, if you have already looked into it, you already know this, but the company is pretty new. It's only about like 3 years old. Technically, it's 4 years old, but like getting the space, getting activities started, whatnot, took another year. So from an activation standpoint, the company is only about 3 years old. So we are kind of at the cusp where we are trying to raise private funding through venture and angel investments. But so far, the company's fundraising has heavily involved Department of Defense, because they obviously see the benefit, the tremendous implication of a product like this for soldiers, 90% of whom actually die of what you just said, injuries, and a big portion of that is preventable if you could take a blood product there. But if the blood product is not there, which is the case right now, if you could take a technology that's kind of like blood products, so we call it blood surrogate products, as a bridge, that improves the survival probability, right?
That's exciting. So how was this, like, since we are coming to the point of your company, like Haima Therapeutics, how was this conceived in the beginning? What were the initial days of Haima Therapeutics?
Sure, sure. This is very exciting for me to talk about because it involves one of my former PhD trainees who is now the VP of research at Haima. Her name is Krista. Well, she was Moderi. Now she's Krista Pawlowski. So Krista was an undergraduate student in Case Western Biomedical Engineering, doing research in Cleveland Clinic, in a laboratory in Cleveland Clinic. And one of her friends was a researcher in my lab. And this friend, Sarah, she approached me one day and said, okay, I have this friend named Krista. She really wants to do, biomedical engineering research. She's looking for a lab. Do you have? And this was like very initial stages, very stressful periods of, junior faculty life.
I can imagine.
Yeah, as everybody will tell you, right? So 2009, around 2009-ish. First 3-4 years after I started as a faculty. I started in 2006 as a faculty. 2003, I started as a postdoc and then transitioned over. So Krista joined my lab and she was brilliant. She is brilliant. So she is one of the people who kind of, got involved in this artificial platelet idea that I tried to sort of Jedi mind trick into people that had just joined the lab. And there was another trainee, Madhu Ravi Kumar, who ultimately moved to a different lab and got her PhD in neural engineering area, neuroinflammation area, and is working in a company currently in that area. So Krista and Madhu were the first two people that sort of ran with this idea. And obviously the idea went through many iterations. Krista graduated as an undergrad and wrote an NSF graduate fellowship application, which she got funded the first time. And she graciously decided to continue her PhD in my lab. And for her PhD, she picked this artificial platelet topic as a central theme. And so it was a lot of, heavy legwork from her that got many of these iterative versions of this artificial platelet product. What you see today was not what we started with. It was something similar, but it kind of evolved into a more refined system. So once Krista graduated with her PhD, I asked her, like, what do you want to do now? And because, I asked this to all my trainees so that, I can help if possible. And she said, well, I really want to go the translational route, but I know we don't have the framework to start a company right away. We could write this grant. And she picked up this grant called the SMART grant from NIH that allows some translational work to be done at the NIH, actually. You send samples to the NIH and they do some evaluations kind of stuff. And she conceptualized the formation of this Haima Therapeutics.
How was the name conceived? Like, who coined the name?
So we looked at, okay, so this is a funny story.
Yes.
Okay. Somebody told us, like, oh, you want to start a company, you should pick like a Greek or Latin name because those work really well. And I'm like, really? Okay, fine.
Apple doesn't sound like a Greek name.
Apple doesn't sound like it. or Google, I don't know. Yeah. But anywho, if you look at many pharma companies, they have some Greek or Latin or whatever. So we looked at all the sort of Greek, Latin names of blood. So sanguine is blood, sanguination. So Haima is the Greek name for blood. Okay, so that's where the word hemo comes from. And so I think I brought that word up, and I asked Krista and some others to kind of see if this is already taken. And to our surprise, it wasn't taken. So we are like, all right, we are taking it because we're gonna do blood-related research, and so this would be perfect. So that's how it kind of happened through brainstorming. It wasn't some strategic, like, okay, let's spend days and days thinking about a name. But it worked out. And then I think back in 2017, 2018, one of my PhD trainees, who is going to join Haima in probably a few months, Ujjal Sekho. So Ujjal and I started messing around with a logo, and he came up with the logo that you see on Haima today. And we had little tweaks and colors and angles and whatever. But Ujjal, I would give Ujjal the credit for the logo. So I'll give Krista the credit for the concept of the company. I have been involved in every way as a co-founder, co-inventor, whatever. But without these trainees feeling passionate about translation and taking up the heavy legwork that's involved in some of this stuff, the company doesn't happen. So I completely owe it to the trainees for the success of the company. And obviously then after the company was formed, the people that we have recruited in the company, the current CEO, Mike Brookman, the current scientists that work there, Emily, Sana, Srijal, etc. It's one thing to kind of start something. It's another thing to sustain it, right? So exactly. I'm just excited to see that this team of people are remaining passionate about the vision of the company and putting in the work to make it what we envision it to be.
I want to congratulate you on the Haima Therapeutics, and the process seems very fast now. I think you guys got a lot of funding recently, and I wish you all success in that arena as well. So one of the last points, since we are running out of time, so I'll just ask one of the last.
I have another 15 minutes.
Oh, is that perfect? Then I have a lot more questions. So in the previous part of our conversation, you had mentioned like your influencers and the mentors and the people who influenced you to take this career path towards the blood-related technologies. So, but a lot of time the name of the Cleveland Clinic and Case Western Reserve University came up. So do you think there's anything unique about the Northeast Ohio or even Cleveland area that helps you to pursue this translational research?
Right. No, that's a great question. And I always say this because I think it's one of the reasons I kind of stuck around Northeast Ohio, although I interviewed at other places and I kind of came back. It is the proximity of medical institutions. You actually get the feeling of patients being treated beyond the science of things, right? That everybody is doing a lot of really cool science all across the board in all these institutes, right? Cleveland Clinic, Case Western, the VA hospitals, university hospitals, Metro Health. There's a lot of really cool science and engineering going on. But all of these entities have a heavy presence of patient care. And so you kind of see that language, that vibe, that philosophy around you, and it drives you to think like that, right? It's very difficult for us to kind of come up with an idea and not think, okay, can it go to a patient? Because you are seeing patients and patient care and clinicians all around you all the time. And so I think it's sort of kind of organically gets ingrained. And I wouldn't say Northeast Ohio is the only place where this happens, but I would say that there are very few places that this happens. And usually the places where this happens are places where science and engineering departments are surrounded by really translational, high-impact patient care institutions. So if you can think of Boston, okay, you can think of Houston, you can think of the Research Triangle, so Duke and UNC and the surroundings. So I can name a couple of different locations, but I can name them on my one hand. Mm-hmm. There are not many, probably 5 or 10 at most.
Yeah, it is exciting that like in that when you go to the Cleveland side, you can see the Case Western or UH and the Cleveland Clinic to be under almost in the same circle. Like, and there's a lot of collaborative works are being going on in there, which is very exciting even for me. Even though we are in Akron, but it's not too far from
Right.
Cleveland area. Yeah.
Right. So I think that has had an impact because it sort of instills this patient care philosophy in your mind, and so everything you do, you try to connect it to patient care, and then make very informed decisions that okay, I'm going to do this for the fun of it, and I'm going to do this for translating to a patient, and you could do both, right? But it allows you to kind of make those distinctions.
Yeah. So I want to switch gears a little bit right now, and like then let's talk about some of the career trajectories that you had. So you
Sure.
have received your PhD from University of Akron, where most of the students goes towards an industrial positions or in towards industrial positions. So how did you persevere during this process and went against the odds to pursue a career in academia? Do you have any tips for other budding scientists who want to achieve the similar path?
Right. I think first and foremost, it's not like I set out to go into academia. I will honestly admit that. I did work in the industry for little stints like Procter & Gamble and Guilford Pharmaceuticals and so on and so forth. But at the time that I was graduating, so there were two sort of concomitant factors. At the time that I was graduating, there weren't a lot of R&D going on in the industry. And I didn't want to join a non-R&D sector in the industry. I was not very interested in it. And the reason for that is this was like post 9/11, right? So it was within a couple of years of 9/11. So, and from an investment standpoint, many of the industry investments scrapped R&D programs and were not recruiting or growing their R&D sector. So the opportunities in the industry were limited. So that is obviously one factor, right? The other aspect is more of my personal aspect, that I have always liked teaching, and I have always liked so at Akron, I actually taught the biomaterials core course that I think Dr. Yoon or someone is teaching now. But long time ago, Dr. Stan Ridgers, Ridgers, and there was somebody else. We were actually teaching it. So I was not a TA. I was actually a co-instructor of the course, and I really liked that experience of, inspiring younger generation to do research, think, design, and things like that. So I had that in me, just didn't know that I would kind of pick that as a career, but it was sort of innately present in me. And then I also felt enthusiastic about learning what biomedical engineering actually is in terms of, going into patients. Because although I did my research at Akron in a biomedical field, it was mostly materials design, and it didn't really evolve into getting into, on the biological side of things. And so when I looked at those two factors, I thought that, postdoctoral experience, I still hadn't thought of academic, but I thought, okay, let me see what it is, because postdoctoral experience is a little bit different than doing PhD, because you get to learn management skills, right? You have people working with you or under your mentorship. You get to expand your expertise beyond what you have done, that kind of a thing. So when I interviewed for postdoc positions, I was very consciously not interviewing for a project or for an area where it would still involve making new materials, because I have already done that. I was looking more for applicative, still making materials, but there's an applicative end of it that I wanted to learn. And I interviewed at a bunch of different places, and I chose Case Western because I got excited by this idea of bio-inspired materials design that was involved in making those coatings for blood contacting devices. So that's how I kind of made the transition. So tips for people who want to do this: it's really, I mean, in retrospect, it's really important to be passionate about research, right? Number one. Number two is it's really important to kind of look about 5 years ahead, about 10 years, 5 to 10 years ahead, to see, what do you really want to achieve? Do you want to do discovery? Do you want to do translation? This is not easy to do. I mean, I'm kind of saying all of this in retrospect, but I'm sort of assuming that these things were sort of subconsciously present in my mind, and now I can recognize them looking back, right? Academic job is very challenging, especially the first 5 years or 6 years or so, because you have to do teaching and research and managing things and whatever. Like, it's a multifactorial challenge. And you have to kind of, I learned not to get frustrated by things and kind of put my head down. It's okay if you consciously develop a tunnel vision for a small period of time and then take your blinders off and kind of diverge into multiple projects. But, focus on maybe 2 or 3 specific things that you want to do and kind of go at it. The other really, really important thing is that, and it's true probably in any career, but I can only talk about the academia, is that there will be a lot of naysayers. There will be a lot of people who would maybe underappreciate some of the efforts. If you get lost in that, it's not possible to kind of stay focused on what you really want to do. So you have to kind of put on blinders and kind of go at it in a really focused way. Then once you reach your ground, wherever you are focused on, then you can take your blinders off, because you will see that all these, paraphernalia, all these outside factors, somehow they have kind of gone away because you put your blinders on. They got tired of messing with you, and
I agree. In a context where your colleagues are going to a different and high paying plus a diverse career, when you're trying to still being in that academic realm, it's hard to persevere in general. I have talked to many people, and they try hard time continuing with that perseverance. Yeah, it is
right.
It is good that you were able to overcome all those and to achieve.
I think you hit on the word really, really, really well. I think perseverance, persistence, and patience, I would say the 3 P words: perseverance, persistence, and patience. There is no substitute for it, right? And it is not easy to do. I mean, it's easy for me to say this now looking back, but even now, it's not like, oh, I have like a clear field and I'm running. There is a lot of, it's an obstacle course. Mm-hmm. It is an obstacle course. Yeah. But perseverance, persistence, and patience, it's possible to, I think that's the 4th P word, right? Mm-hmm. It's possible to overcome many of these things.
Yeah, that's a very interesting tip. I think we're running almost in the time. So I'll ask you the last question I always ask everyone. Is there anything that is in your not to-do list, which you don't do because you are in the position that you're right now?
Scientifically or philosophically? Or?
Philosophically would be the best answer, I think.
I try not to get involved in negative criticisms, right? I am very okay to say things like, I don't know, right? It is important to recognize what you do know and what you don't know. Because, I would see a lot of noise. That's what I would call it, noise about, people already. So there's this thing we always talk about as implicit bias, right? And people would get sort of caught up in that. Reviewers do, researchers do, faculty do, everybody does, right? If you go at that with that mindset, you have already lost reading half the story, right? And I try not to do that. I'm not saying that I don't sometimes end up by mistake doing it, but I try my level best not to do that. I take everything as it comes. and make that a learning experience for me, or try to make that a learning experience for me. And then because of my knowledge or expertise or experience or whatnot, I may find pro or anti arguments to what I'm looking at. But I try to not delve in negative criticism, which essentially means I try to give as much as possible constructive criticism. So you can still criticize something, but constructively. So there's number one. Number two is I don't really get hung up on impact factor and how many. I mean, yeah, if we get a high impact factor publication, I will talk about it. Mm-hmm. But like, I don't make that my desperate goal in life to be nature or nothing, or science or nothing, or whatever it is, right? To me, the impact that I think of is I want to get into a patient. I don't care if I have high impact publication or not. I'm going to move the needle as much as possible to try to get, that is more exciting for me, right? So I don't get lost in impact factor and followers. And, it's actually funny that people I saw on Twitter after this whole Capital Chaos fiasco, or it's all this conversation on, I lost 800,000 followers or whatnot. Like, I don't even count who is following, who is not following, because, whatever it is. But I guess I don't get lost in the, try to not get lost in the glamour of things, and try to keep my focus on the usefulness of things, right? So this is number two. And number three is I do not make any research or scientific endeavors transactional.
What do you mean by that?
Yeah. What that essentially means is this is why I think I have been, at least so far, very successful at collaborations or building collaborations. Because I never go at anything as, okay, I will do this for you. What can you do for me? That's a transactional relationship, right? And nothing wrong with it. Lots of successful things happen through transactional relationships. I try to not do that. I try to always make things as our thing. What can we do? Where can we go? What can we reach? Where can we advance? So this feeling of we, the team feeling, is really important to me. So I try my level best to not get into the here is what I will do for you, so you should do this for me kind of thing. That is present a lot in academia, and I'm sure outside academia too. So those are the three things that I can think of philosophically that I really consciously don't do, or try to do as minimum as possible.
Yeah, I think these three are very important points. I think there's a deflection in the academic point of going after the implicit bias, or even having the transactional relationships. So I think that probably is the reason why you have been very successful in collaborating with people, because I have seen your research and most of it are collaborated across disciplines. Like you have worked with clinicians, you have worked with biologists, engineers, and even army personnel, right? So this is exciting. So thank you for your time, and this has been a wonderful talk. And I think I have a lot more to dig deep into, but because of the time, we can have a round two when the time allows. And then, yeah, it has been so much fun. Thank you so much, Anirban.
Absolutely. Before we sign off, I do want to say one thing really, really emphatically and enthusiastically, because I think that this point has, and you probably already talk about this, but this point has to be made because I was talking about team. One really important sort of sub-factor in that team thing is not just cross-disciplinary or multidisciplinary or whatnot, but diversity. It is really, really important to not undermine the strength of that word and the importance of that word. And if you look at our lab as well as our company teams, it's not like a random thing that we try to build a very diverse team. It's actually very consciously, because we want to make an impact societally, not just clinically, not just financially, not just, scholarly, but societally. And if you want to make an impact societally, whatever scientific career one may choose, I think it's absolutely important that you put diversity as one of the forefront impact factors in your endeavors. And so, this is, I mean, I say this for all scientists, that be very conscious of doing this, because this actually helps society.
Yeah, I completely agree with that statement. To have conscious decisions to make sure that the diversity is maintained in the group, or even in the teams that you create. So yeah, with that, yeah, thank you so much for joining me. Thank you so much, Anirban. Thank you for your time.
This is great. Yeah. Thank you.
Yeah.
Have a good day.
That's it for today, guys. I hope you all enjoyed this episode. Please find the show notes at thesoftmattershow.com for more details. That is thesoftmattershow.com. You can write your questions, suggestions, and everything else to me at Amal Narayanan at thesoftmattershow.com. See you with another exciting episode soon. Until then, goodbye.